Thank you very much, and it's an honor and a pleasure to sort of kick off this meeting with a talk about our rapidly deepening understanding of the molecular heterogeneity of this disease. And in some ways, I think that part of the point of this talk is actually the fact that our way that we classify melanomas is evolving. It used to be it was strictly based on anatomically where the tumors arose from, and we're now moving into a molecular era of having molecularly defined subtypes of melanoma. These are just my disclosures. So for the last few years, I've sort of been putting up this same slide at the beginning of most of the talks I give at meetings and to fellows, talking about the recurrent events that we know of in melanoma that are driving this disease. And clearly the sentinel event in understanding the molecular biology of these tumors was the discovery in 2001 that almost 50% of cutaneous melanomas have point mutations in the BRAF gene. So BRAF is a serine-threonine kinase in what we call the RAS-RAF-MEK-MAP kinase signaling pathway. 90% of these mutations affect a single site, the V600 site, and most commonly result in substitution of a glutamic acid, the so-called V600E mutation, although there are other substitutions that happen at that site. What these mutations do is they take the BRAF serine-threonine kinase that we can normally turn on and off and make it constitutively active. So now this pathway is about 100 to 200-fold activated compared to cells that have a normal BRAF gene. And certainly the clinical results with the BRAF inhibitors, which will be discussed in much greater detail in subsequent sessions, have validated how essential this mutation and this pathway are in the disease. Now, in addition to BRAF mutations, approximately 20% of patients with cutaneous melanoma have activating mutations in RAS in their tumor, specifically in the NRAS isoform. And what we know is that in patients who are treatment naive, these events are essentially mutually exclusive. It's less than 0.5% of patients that will have both of these mutations. If you have a BRAF mutation, you won't have an activating NRAS mutation. If you have NRAS, you won't have BRAF. And it's pretty clear that the MAP kinase pathway is one of the pathways that RAS mutations drive in order to contribute to oncogenesis, but certainly data from multiple tumor types suggests that the RAS mutations activate other pathways as well. One of those pathways, which again has been implicated in multiple tumor types, is shown on the right-hand side of the slide, called the PI3 kinase AKT pathway. And one of the ways this pathway can be activated is actually not by an activating mutation, but by losing the tumor suppressor PTEN, which is the major negative regulator of the PI3 kinase pathway. We see loss of PTEN in 10 to 30% of melanomas, and that actually correlates with constitutive activation of the pathway. Now, getting back to the genetic patterns, in general, PTEN loss is mutually exclusive with the presence of an NRAS mutation, the same relationship we've seen between BRAF and NRAS. However, many of the tumors that we see that have loss of PTEN have a concurrent BRAF mutation. And so just summarizing those three events, really just looking at those three oncogenes, what it's pretty clear is that we have a very high prevalence of oncogenic mutations in the MAP kinase pathway, particularly in the cutaneous melanomas, and evidence from the genetics of this disease that at least a subset of patients are activating multiple pathways at the same time, again, based on a very simplistic understanding of what are some of the molecular changes that happen in this disease. So in 2009, many of the speakers, like myself, were in the audience at the Melanoma Congress meeting when Michael Stratton from the Sanger Institute presented the results from the first case of melanoma that had undergone whole genome sequencing. This was a tumor, actually a cell line derived from a melanoma tumor from a 45-year-old patient. It had, again, that common BRAF mutation, loss of PTEN, and also loss of P16 encoded by the CDKN2A gene, which is the most frequent event we see in familial melanomas as well. And the stunning finding that was presented very early in the presentation was that in this initial sequencing event, they showed that the tumor had over 33,000 changes compared to the normal DNA from this patient. Most of these changes, again, reflected the influence of ultraviolet radiation. There's characteristic mutations that we get. And again, just getting to the complexity of this, over 180 of these mutations were in the coding regions of genes, and so it was predicted that essentially there were almost 180 different mutant proteins that were now present in this patient's tumor. And so the daunting task is going to be, with all of these mutations present, how are we actually going to filter that information down to figure out what of these events is actually important to us clinically, either as prognostic factors or, again, as therapeutic targets. And certainly with a high mutation burden like this, one of the first priorities then was to actually be able to get data for many different patients to understand what are the recurrent events. And so over the last several years, we've seen small studies that have been published with a limited number of patients who have undergone whole exome sequencing, confirming that we're dealing with a high mutation rate. But what really has been the breakthrough is in 2012, we had 2 large sets of whole exome sequencing data from cohorts of cutaneous melanoma patients. And what we're moving forward with now is the fact the melanoma TCGA data is starting to be released and likely will be published early next year. And so what I'll do over the next several slides is really focus on these studies in particular and what we've learned from them. So the first study was published by Ruth Halaban's group at Yale. They presented the study of whole exome sequencing of almost 150 melanomas. When you actually start looking into the details, actually it's 100 of these where we had not only DNA from the tumors, but also for normal DNA, so that we could actually distinguish what were really somatic events as opposed to potentially polymorphisms. And it actually is about 2/3 of those samples were tumors, and about 1/3 of them were actually short-term culture cell lines derived from patient tumors. One of the things that's interesting is that, again, although about 100 of these tumors are actually cutaneous melanomas, this is one of the first times we actually saw sequencing data from other melanoma subtypes, including the acral melanomas that are all in the palms and soles and under nail beds, and mucosal melanomas, both of which are tumor types where the role of ultraviolet radiation is much less clear. Overall, again, supporting the mutagenic effects of ultraviolet radiation, the tumors that were, again, that were sequenced, that were cutaneous, had on average 171 mutations present in each of those tumors, compared to only 9 in the tumors that arose from areas without evidence of sun exposure. And this is a slide that Ruth Halaban has shared with me and with other investigators, summarizing the main events that were found. And one of the first take-home lessons is that by far the most common events that we're seeing, particularly in terms of recurrent mutations, again, were the activating mutations in BRAF with the V600 mutations and in NRAS, which cluster at G12, G13, and Q61. For anybody who happens to be an aficionado, one of the things that's interesting about melanoma is that in other cancer types like colon cancer, the mutations tend to cluster in the 12 and 13 region and are rare in 61. Melanoma is the inverse of that. 80% of our mutations happen at Q61. But to date, we don't have any evidence that there's a functional significance of having a mutation either in exon 1 or in exon 2. A number of other genes were identified as being significantly mutated, including several which are known tumor suppressors, but a variety of genes that are potentially novel tumor suppressors or oncogenes. And the one that was a particularly striking finding was the identification of recurrent mutations in a gene called RAC1. RAC1 is a small GTP-binding protein that's a regulator of cytoskeletal rearrangement, and thus it is involved in cell motility and invasion. While all of the mutations, again, in BRAF and NRAS tend to cluster around several hotspots consistent with the functional significance of those events, the mutations in the other genes are spread throughout those genes, which is okay for loss-of-function mutations. But again, the striking thing was that the RAC1 mutations happened over and over again at the same site. The other thing that's interesting about this is that although we talk about ultraviolet radiation being very mutagenic in melanomas, in actual fact, the mutations in BRAF and NRAS don't have the signature of a UV-induced mutation, whereas the RAC1 mutations do. This actually appears to be our first clear-cut evidence of a hotspot mutation that's actually induced by ultraviolet radiation. So they went on to do functional studies to try and see whether we could really understand the significance of these mutations in RAC1. What's shown in the upper right is biochemical evidence that when you put this mutation into the normal gene, it has more biochemical activity. And down at the bottom, it impacted the growth of cells. They grew slightly faster, and then in particular, they actually migrated much faster than cells that were either having just their endogenous RAC1 or overexpressing a wild-type form of RAC1. What we don't know yet, again, is the therapeutic significance of this. There are RAC1 inhibitors, but to date, there is no information available about the efficacy of those agents, particularly in tumors with RAC1 mutations. Now, in parallel to this study, there was a second paper that was published by the groups of Levi Garraway and Linda Chin, whole exome sequencing. This is now 121 tumors, all of which had matching normal DNA. So more tumors where we actually absolutely know which mutations are somatic as opposed to polymorphisms. Of note, though, 76 of these samples were, again, actually cell lines that were derived from tumors as opposed to being clinical specimens themselves, and this was overwhelmingly cutaneous melanomas. And again, just to give you an idea of the burden of mutation we're talking about, more than 13,000 genes showed evidence of a somatic mutation in at least one tumor. And if you said, well, I only want to know about genes that were mutated in at least 10 tumors, that list is still over 500 genes. Again, reinforcing the challenge we have moving forward in understanding what the real drivers that there are in this disease. Interestingly, their analysis actually showed that certain regions of the genome are more prone to develop mutations than others, leading to new bioinformatic approaches to analyzing the data to, again, try to separate out which genes were most likely to be driver mutations as opposed to passengers. And this, in the end, led to the conclusion that there were 11 genes that were significantly mutated. And so this actually, again, used sort of a new analytical approach, and that may actually be the reason why there is some differences between the results here versus the Halaban study. There were 6 genes that were identified as significantly mutated, which, again, we already knew about as being recurrent events. Again, BRAF and NRAS were most common. And then there were 5 genes that were purported to have been novel targets of somatic mutations in melanoma. This table is meant to really be presented as a reference. These are the genes that, again, are the potential novel targets of mutations in this disease, the majority of which we really still don't know the functional significance of the mutations that we've actually found. As summarized from the previous paper, we do have functional evidence to support the RAC1 mutations truly affect the function of RAC1, but we actually don't have functional data for these other mutations yet. Some of the genes, like STK19, which is a kinase, show that many of the mutations do actually cluster to a certain hotspot or hotspot region, but again, we see mutations elsewhere in the gene. One of the other findings was that there was a significant rate of loss of function mutations in a gene called ARID2, which is involved in the chromatin remodeling complex and brings sort of the idea of epigenetics being a significant target of mutational events that we see in melanoma. And this is actually a recurring theme we've seen as we've done whole exome sequencing across multiple tumors, that there seem to be multiple mutations in that gene family, and melanoma certainly falls into this. This is what I think is really the iconic figure that actually has come out of the 2 papers, and this was called the landscape of driver mutations in melanoma. So on each row, we see one of the genetic events we see. The upper half above that solid line, again, are the actual mutations that were found in genes. Down at the bottom, the investigators went back and did assess both gene deletions and amplifications of certain oncogenes and tumor suppressors, which had previously been implicated in melanoma. And then looking up and down, each column represents a single patient. And so again, what we can see, and here, I'm sorry, I'll use the pointer, is that the most common mutations we're seeing were those hotspot mutations in the BRAF gene, which are in red, and the recurrent hotspot mutations in NRAS. But certainly what we can identify here is that when a patient has a mutation in BRAF, they likely also have multiple other events which may be of significance and may contribute to the heterogeneity that we even see among patients with these driver mutations, and similarly for NRAS. Now, certainly one of the questions we've had going, I'm sorry, how do I go back? One of the things that we'll come back to is what these studies are also starting to present data about is what are the drivers that are going on here in these tumors that don't have BRAF and NRAS mutations. Now, a third study, which is really sort of, again, still in progress, is the melanoma TCGA effort, which overall is aiming to do molecular profiling on 500 melanomas. The goal is specifically for this to be done on cutaneous melanomas and not on other subtypes, and actually, due to the amount of material that was needed, the goal really was to focus primarily on regional metastases, either lymph node metastases or subcutaneous tumors. However, there was also allowed the possibility of including primary tumors from the same patients as well as distant metastases from the same patients. Now, one of the real opportunities we have from this is that we're actually doing multimodality profiling. So in addition to all of the tumors undergoing whole exome sequencing, a subset of them will undergo whole genome sequencing. All of the tumors are also being analyzed for changes in DNA methylation, mRNA and microRNA expression, and the expression and activation of protein signaling pathways. And all of the samples will also have clinical data associated with them. So as opposed to the other two studies, which sort of gave us rates and prevalence and patterns of mutations, this data set will allow us now to start integrating what other changes correlate with the presence or absence of specific mutations. And so this is actually the initial public report of the data that was presented at the TCGA workshop late in 2012. This was about halfway into the study, a little over 200 tumors. And in actual fact, the mutations that have been reported so far are basically the same as what has been reported in the other studies. I think one of the things is that the two studies by both the Yale group and by Linda Chin's group have really actually given us a pretty good list of what the mutations are. Again, the most common ones being in NRAS and in BRAF. But now what's starting to appear is what we think is a distinct or third subgroup, which are patients who have mutations in the NF1 gene. NF1 is a negative regulator of the MAP kinase pathway, specifically of RAS, and so it gives us sort of another way in which this pathway can be activated. And notably, this is a statistically significant enrichment in the patients who have a BRAF-NRAS wild-type tumor. So focusing in on those tumors, again, that don't have the hotspot mutations in BRAF and NRAS, in the Linda Chin study, this is 20 tumors in TCGA. In that initial report, it was about 50 tumors. What are the events that we're seeing in those patients? And so again, as I've highlighted, one of the events that seems to be particularly enriched here are these loss-of-function mutations in NF1, giving us another way to activate the MAP kinase pathway. And in actual fact, when you look at these genes, what we see are multiple alterations that, again, probably result in activation of the MAP kinase pathway. So even though these tumors don't have the activating mutations in BRAF or NRAS, the data supports that nearly every melanoma has something that drives that same pathway. I think the other commonality is that most of these tumors also have some type of aberration in the genes that regulate the cell cycle. Cyclin D1, CDK4, and CDKN2A, or also P16. And again, these findings have therapeutic implications for the development of rational therapeutic approaches for these patients. Now again, most of the focus to date on melanoma has really focused on the coding regions of these genes. However, two different studies were published back-to-back in Science earlier this year, showing that there can be significant events that happen even in regions that aren't in the coding regions of genes. And actually what these two groups both found, one in familial melanoma and another looking at somatic events, were hotspot mutations in the promoter of the telomerase gene. Telomerase is a protein that's needed in order for cells to be able to survive multiple divisions. It's something in preclinical models we often sort of induce this artificially, but this is now looks like the mechanism of how this actually happens in patients. So as I talked about, hotspot mutations are things that happen recurrently and usually are functional. So interestingly, what were found were two recurrent hotspot mutations, not in the gene itself, but in the promoter region. And actually, if you add these up in this series of patients, it actually ended up being over 70% had at least one of these two events. And what these mutations do is generate a novel binding site for a new group of transcription factors. And when we do this in the laboratory, it results in increased transcription and therefore increased expression of telomerase. Other events which are also we're starting to understand are gene fusion events, or fusing different parts of the genome together. Boris Bastian presented data at ASCO that's recently been published, I understand, showing that in Spitz-Nevi, a relatively rare subgroup of melanomas, but ones which don't have BRAS or NRAS mutations, that over half of those tumors have genetic fusions that actually bring together receptor tyrosine kinases and other genes, which appear to actually result in activation of those genes. There is some data that BRAF fusion events can also happen, although they haven't been characterized extremely well in melanoma, although there have been some reports that have been given at meetings suggesting that this may be another event that happens in the tumors that lack hotspot mutations in BRAF or NRAS. And so then just to put the information together about what do we know right now about the recurrent mutations in melanoma, and specifically focusing on cutaneous melanoma, is again just emphasize this has really been a very rapid evolution in our understanding. It's really just over 10 years ago that the BRAF mutations were reported, and we started to understand that the MAP kinase pathway was a critical player in this disease. So 10 years later, we really have our first true set of data that we can look at for whole exome sequencing, and 2013, 2014 is when we'll get the data from the Melanoma TCGA project. Going back to what we knew initially, the most common mutations remain those activating mutations in BRAF and NRAS, but we're now starting to get an understanding of what potentially the heterogeneity and drivers may be of those tumors that lack those hotspot mutations. And again, as I've shown you, the pattern we're seeing are frequent mutations in genes in the MAP kinase pathway, particularly NF1, and also regulators of the cell cycle. And the question is when we find all of these different mutations, is how are we actually going to decide which ones of them are actually important? And I would argue we have a number of different approaches that we'll be undertaking over the next 5 to 10 years. So first, the TCGA data is an opportunity to integrate the mutational data with other molecular platforms to understand if there's other molecular changes that we see happening when these mutations are present. Certainly in the laboratory functional testing, as we've described with the telomerase mutations and the RAC1 mutations, ultimately for patients, what we need to know is which one of these mutations will help us guide the therapy and improve outcomes in patients. And that could include tests that are prognostic, helping us to understand which patients with early stage melanoma are at high risk or low risk for relapse, and certainly whether any of these can actually be therapeutic targets, like we've had the experience with the BRAF mutation. And again, just with the telomerase mutations as an example, we really haven't started digging into the significance of those mutations that do happen in non-coding regions, which again is a daunting task because of how many of those mutations are present. I'd like to highlight on this slide, I think, a couple of the other challenges that we have moving forward. So one of the questions is molecular heterogeneity. So with the activating BRAF mutations, we know that these are not only the most common somatic events that happen in melanoma tumors, but they're also present at a very high rate in benign nevi. And we've been able to show in multiple different studies that if you look at different tumors in the same patient, the BRAF status is remarkably concordant. However, there are other alterations where there appears to be discordance, and for the majority of the mutations I presented today, we simply don't know whether they're concordant or not. What we're also starting to see is the concept of heterogeneity even within an individual tumor. And this is actually down here an illustrative case that actually refers to a couple of different challenges. This is actually a tumor from a patient with an activating BRAF mutation who progressed on a BRAF inhibitor. An analysis of this progressing tumor showed that part of the tumor had marked reactivation of the MAP kinase pathway despite continued treatment with the BRAF inhibitor, and this part of the tumor had not only a BRAF mutation but also an NRAS mutation, whereas another part of the tumor hadn't really turned the pathway back on and didn't have an NRAS mutation. And this links to one of the other challenges we have moving forward, which is the understanding now that the treatments we give can actually change the molecular biology of these tumors. As I mentioned, in newly diagnosed treatment-naive melanomas, NRAS mutations and BRAF mutations are mutually exclusive. But in patients with a BRAF mutation who've been treated with a BRAF inhibitor, at the time of disease progression, what happens in almost all of these patients, 20% of them have now both a BRAF mutation and an NRAS mutation. And so certainly moving forward, we'll want to know about the molecular biology of newly diagnosed melanoma, but also a real need to understand potentially the molecular biology of tumors that develop resistance to new effective therapies, including not only targeted therapies, but I would also argue with the immunotherapies. And finally, I think this links to Sansi's next presentation is, again, we've really been focusing on the role of these mutations in advanced stages of disease. We need to understand what their role and clinical significance is in early stage disease. And again, this is a disease where there's such an opportunity actually for prevention and where that could really have ultimately the greatest impact to understand, do any of these mutations or their related pathways have significance to understanding how melanoma develops at the beginning, and are there steps we could take to prevent or reduce that? So with that, I'd like to acknowledge, again, the members of the Melanoma TCGA group and multiple people who were generous to share slides, and I'd be happy to take any questions.